pleasure to introduce my good friend, my long-term collaborator, Professor Omar Farha. He's been the prolific scientist in the field of MOFs. He's one of the two Omars that built the field, along with other scholars in the field. And of course, it couldn't be any better at this time of the year, because this year was the MOFs year for Nobel Prize in Chemistry. I think this really adds tremendous value to this lecture, which is already a very amazing talk, which I've seen Omar talk a lot. So I really enjoy every time I've been inspired a lot. Of course, he told me keep it only 30 seconds introduction because he has a lot to show, so I will try to keep it short. You can Google his long CV and accomplishments, but just to give a taste that he has more than 700 papers published, and he has all the metrics anybody would need. But what's so striking about him, which I really am a big fan of, is there's an award made in his name at Northwestern University. So it's not that common to a lot of living professors and scientists to have an award in their name. And it was established because of his strong safety consideration. It's both scientific merit as well as his strong safety cautions. These are really important for me as a chemist, because we want to do a safe science. And of course, you can get better idea for Omar's astonishing career online. But when I asked ChatGPT, it said to me what I already know. He's the most prolific scientist in 21st century, and I strongly agree. But of course, you will get to say what he has to say in MOFs field. And please welcome Professor Omar Farha.
Thank you. Can you guys hear me? Awesome. Thank you, Jafar, for a very, very generous introduction. And thank you all for being here today. And thank you for inviting me once again to KAUST. I consider KAUST is my second home, and I love it to come here. So before I start, let me actually give my thanks. And the biggest thanks go to the team that actually does this work. I am here just speaking on their behalf. And this is the current picture of my group right now. We took this picture a few months ago when it was really cold in Illinois, and it's cold again. I left while it's snowing to come to beautiful, the kingdom here with the weather is awesome. And you couldn't do hard research if you don't collaborate, and I collaborate heavily with my colleagues. When he was a professor, he's now not in academia, Professor Kramer, but Snare, Galdradi, and Professor Hub are, a long-time collaborators. And more importantly, the people who I will be telling you about some of the results they've been given to us, and they've been a long-time collaborators as well. For example, Dr. Peterson, we've been collaborating for 16 years right now, and we published more than 70 papers together. And you will see why, because the materials I gotta be telling you about, nobody should be touching in academia, and the professionals are the only ones should be doing this. But you'll see how we do it in academia in a university setting, and then what's the professionals do. And the funding agencies, without them, I would not be here to be able to tell you about what we do. So the main thing, my group is really a synthetic group at heart. We work under the area of energy and environmental science. We like to do catalysis, storage, and separation. And for today, I want to walk you through one project that it took us about 15 years to get to this point, from PowerPoint chemistry all the way to a commercial product that people are using and utilizing. And it's gonna encompasses actually all three areas: the catalysis, the storage, and the separation. In my group, there is many active projects, but for today, we will concentrate mainly on one, which is human protection. And for today's talk, human protection, I will start with talking about military uses, But you could very quickly, because that's who pays for it right now, but very quickly you could see that this is going to be going into the firefighters, the farmers who are spraying these chemicals, all the way to the industrial workers who need the protection gear when they're dealing with very toxic chemicals. But for today, we got to concentrate on the DoD type of applications. And typically, people talk about outlines. I'm going to give you a timeline. In 2010, that's when I got the first idea to do this. In 2012, we got funded by the Army labs. And it took us about 12 years for some of those soldiers to be wearing the uniform that actually enabled by the technology we did at Northwestern University under this umbrella. And these are the chemicals we're going to be talking about. And you could see from the names, I don't need to name them, that some of the most toxic chemicals anybody can encounter. And the reason for that, we all as humans, we have this enzyme called acetylcholine esterates. Its job to do the reaction that it's showing in the bottom of the slide, take this neurotransmitter and cleave it in half for a particular function to stop. So give you an example. Right now, I am changing the slide. So I changed the slide, but I stopped. I didn't keep changing the slide because that enzyme did its job. Went and cleaved that messenger, and I stopped doing so. However, if these toxic chemicals are there and they go bind to one of the amino acids where that catalytic reaction take place, now this enzyme is unable to do its job, and very quickly it leads into really not good outcomes very quickly. So here, what I'm showing is protection gear is not a new topic. This is pictures from more than 100 years. But the concept I want to talk about today is these canisters that right now these soldiers are wearing, and whatever these fancy soldiers they are wearing right now, I would argue more or less is the same, except the engineered materials inside them is different. But the baseline of the material is exactly the same, and it has something in common with what most of us have in our kitchens, which is activated carbon. And I always say nothing wrong with activated carbon. I think it's a great material. And what we got to talk about today is a complementary material to activated carbon. But there is a very particular differences between activated carbon and the metal organic frameworks that I want to talk about today. First, the first thing, ignoring the structural motifs, we'll talk about this later, activated carbon has the ability to only capture these toxic chemicals, and that's it. You capture them, whatever is been captured inside those activated carbon stays as a toxic chemical. So the idea was, can we actually start thinking about the new technologies that you have the idea that you could separate, get the toxic chemicals out of the air, store it, and detoxify it instantaneously. That means what's inside that mask or that garment is now safe to handle instead of an exact, still active toxic material. And we knew that way back in 2010, that new materials need to be discovered. And as I said, it's not just for the soldiers, but there is farmers all over the world that they spray toxic chemicals. And I will be showing these chemicals. We use them as actually the surrogates in our lab. Those are actually the pesticides and insecticides that actually the farmers are spraying day in and day out, which means they should be able to use this technology. As I said, right now, the next projects we're working on, firefighters, when they made masks and suits for them, they were done in the 60s and the 70s, and the technology never changed. If you think about the houses, at least in the US, in the 60s and the 70s, they were made out of wood. And those, when wood burns, there is a specific chemicals that comes out of it, and the suits and the protection gear for firefighters was made for that. If you come to the US right now and you see what the houses and what's inside the houses are made of, it's every compound you could imagine, and those suits and those masks are not meant to be for that. And that's why, at least in the US, the average age of a firefighter is 55 years. 1 every 3 firefighters will have cancer in their lifetime. And the reason for that, because even with their protection gear, that's not good enough. And hopefully this technology can be able to help in the future. And from the title, is no surprising to here that I will be talking about metal organic framework, and this is the mecca for that. The Adaudi group and many other groups establish what we all stand on their shoulders for many years. But I will tell you that why I decided to do that for my career, because these materials are programmable and scalable, and hopefully by the end of the talk, I will convince you. You could tell me, is 300 metric tons per year scalable or not? And we could happy during the Q&A to decide, can we go higher and what's the showstoppers? More importantly, there is, the optionalities of having, at least on a computer, millions of these materials, and there is about 25,000, 30,000 already made, because you have a whole periodic table to pick from, the cheap, expensive, and anything in between, and you have simple metals, and you have simple ligands as well, and complicated ligands. What is that gonna give you is, once again, the optionality that you could make a lot of materials with different morphologies, different topologies, and different chemical and physical properties. That means, if you really wanna go after really hard applications, you don't need to settle for only 6 materials out there and pick one from. You could actually go and design a material to fit your applications precisely, and we'll show some of these applications. But the main thing is, just to make it easy for the rest of the talk, I'm not gonna be showing crystals. I wanna be showing the smallest repeating unit, but for the rest of for the audience here who are not, they don't work with MOFs, just these materials, that's small repeating unit can go in the 3-dimensionality. But at the end of the day, that's the nano regime and that's the nano cavity. That's where really the action take place and where our catalysis take place. More importantly, the difference between homogeneous and a heterogeneous molecules or materials, in a homogeneous world, you have a small molecule, you could go get an NMR, you could get a mass spec, and you know exactly what you have. Most heterogeneous samples are amorphous, if you're talking about activated carbon, and we hand wave exactly what the structure, and we've been doing okay with that. The question becomes where MOFs can fit. It's really, I look at them as the borderline between a homogeneous and a heterogeneous. It behaves as a heterogeneous material, but we could manipulate it as a homogeneous molecule and get the structure down to the atomic level, and I will show you some of these materials and how we think about it. If you're talking about masks and suits, things have to be light. You might say, oh, what's another half a pound, or what's another? No, everything has to be light. It has to be very thin layers. And that's why it's very hard to compete against metal organic frameworks, because, for example, if you take NU 1501, 1 gram of that, it's really nothing but the weight of 1 piece of M&M. 1 piece of M&M is really tiny, but that has the surface area of 7,300 square meter per gram. What does that mean? You could talk about soccer, you could talk about the American football, but whichever way you lay it, if you are able to take that 1 gram and lay it on the gram, you could actually cover more than 1.3 football fields, which is closer to about 2 soccer fields. So that's really incredible. That means with a small piece of M&M, I could really have a very thin layer inside my mask. My mask does not have to be giant. My suit has a very thin layer. That means it behaves the way a suit should behave. It's not gonna be restricted and stiff. Now let's go back to the chemicals. And I told you we cannot use these materials in a university setting. And that's why we go and we use what we call simulant or surrogate. The DFP and the DMNP, as I said, those are nothing but pesticides and insecticides. So these still, in our labs, we have to treat them with respect. Pesticides and insecticides are very toxic chemicals, but a lot less toxic than the actual agent. The chemical reaction we want to do is this transformation. We want to hydrolyze that PF bond into this POH bond. This might seem really easy, but trust me, it's really complex. You could do it, but it takes time. That means it requires a good catalyst, and you want to do it using humidity in the air, and we'll get to that at the end. Because if you have to use water to hydrolyze these chemicals in a mask or in a suit, you don't have a beaker of water to worry about. You're not doing a beaker, a reaction the way we do it inside the lab. It has to utilize whatever the humidity in the air, and if that humidity is 20% or 80%, this reaction needs to work just as well in both environments. But at the beginning, I will be talking about a beaker of water because when we started this chemistry, we really did not know how to do it and how to do it properly. So we decided to do it the easiest way, benchmark our catalyst, then take the best materials and formulate it and take it to the next step. In my group, we handle this type of catalysis in two ways. The first way, we learn from nature. Nature has some enzymes, and I will tell you about in a second, that they have actually incredible ability to detoxify these nerve agents so quickly. But the problem with these enzymes, they also denature very quickly. And we've been working with the Army labs for the last 8 years with these enzymes, and we are able to stabilize these enzymes inside our MOFs that they could sit on the shelf for more than 1 year and lose no effect. They'll stay exactly their catalytic effectiveness day one and after a year the same. And right now, with collaboration with UT Dallas, I will show you at the end that we're using these materials as injectable for protection. And you will see how they compare if it's a free enzyme versus the composite that we are using. The second way of dealing with these materials is really through being able and inspired from nature, and can we go to the lab and make metal organic frameworks that resembles, I always say resembles or inspired, but I don't use the word mimic for a reason. To me, nature has hundreds of thousands of years on us and perfected a lot of things. I don't know how to mimic nature. I like to be just inspired by nature, and can I learn something from nature to take it to the next step? With the hope, if you learn from nature, can you do better than nature? And I hope I will show you an example of that at the end of the talk. But for today, I want to, or at least at the beginning, I want to concentrate on being inspired by nature. And I want to end with 2-3 slides that a project we've been working on for 4 years. And finally, we just submitted the finding after we did the IP and the patent, showing how these composites can really have some protection for almost 80 days, even under really harsh toxic chemicals, without using protection gear. So the first thing, this enzyme called phosphotriesterase, if we diving into the active site, you will see it's made by two zincs bridged by a hydroxyl, some amino acids, histidine, some carboxylic acids. When we started this, and before I get there, you could see that this is really the mechanism. It's a Lewis acid-based catalysis. And when we started this particular project, zinc-based MOFs, they weren't stable enough back then. That's back in 2010, 2011, to the harsh conditions that we wanted to use. However, zirconium-based MOFs were coming online at these days, just new to the community. And if really zinc +2 is what we need, a Lewis acid, the thought is zirconium +4 should be even a better Lewis acid, but it gives us the stability that we need. And at that point, you get this for some of the people in the audience, very familiar with this cluster, zirconium-4 clusters, and they are capped by 12 carboxylates. That's why it's called 12 connected. Carboxylates with zirconium is quite stable, coordination, and you could really almost boil it in water and it should be fine. You might hear me using the word 8 connected, 6 connected, 4 connected. What does that mean is the number of carboxylate capping that structure. And you might say, how about when you go from 12 connected to 8 connected, what happens with the other 4 sites? They become hydroxyls, chlorides, and water molecules to balance the charges. And we'll talk about this going from 12 to 10 to 8. A lot of people call it defects engineering. I don't think in this case it's defects by design, because literally it's in a crystal structure and every node has the same exact thing. So in that regard, it's a crystalline and it's not just random defects. The first material we dealt with is this MOF called UIO66, and it's built from that cluster and benzene dicarboxylic acid. the reactivity, how we test this reaction, if you take the surrogate, this is our surrogate, and if you do the catalyst with some base, and here we use ethylmorpholine, and a beaker of water, just for the ease for now, we could monitor this reaction because the product is yellow. However, the actual agent has no product that is yellow, so we need to use a different way of testing that, and we use in situ phosphorus NMR, and regardless if you have the in situ NMR or if you have the UV-Vis, you'll end up getting the same results. Going back, our first publication, it took us about 3 years to get it right because the results were interesting that we did not want to publish one paper and retract another, that we published it in 2014, but Mike Katz, he's a professor now in Canada, he worked really hard for 3 years to do that. And what do you see here? That this material is very selective. It does the hydrolysis quite well. And I'm going to be talking about half-lives. Half-life is 50% of the conversion of whatever time. So why I'm telling you about half-lives and not turnover frequency and turnover number? Happy to talk about this during Q&A. But turnover number is just how long have you ran your reaction and how many times. The half-life is the half-life under the same conditions. If you did it in Evanston, if we do it in Jeddah or anywhere, it doesn't matter how long you run your reaction, your half-life stays the same under the same conditions. And the Army labs, they wanted to be able to do it if they do it in Spain, which we had collaboration with Spain, or in the UK, or in the US, that they're all getting similar numbers. So when we did that, actually, one thing to just point out too, the half-life here is 50 minutes. And you might say, okay, is that good or bad? How can I benchmark what does 50-minute half-life means? Before this, I had a postdoc for using supramolecular assemblies that spent 6 months to make 100 milligrams, and the half-life, which was the best back then, 36 hours. And that was the best out there that is catalytic with a half-life of 36 hours. Making 2 grams of this overnight, and after you process the material in 2 days, you have a gram of that, going from 36 hours to 50 minutes, that was huge for us and for a lot of people in this area. But when we send this this material to the Army labs, they get now a half-life with the actual agent of 10 minutes. And you might say, why is it faster with the actual agent than the simulant? Because the actual agent is actually more reactive, and that's why more toxic, versus the pesticides and insecticides. They are less toxic because they are less reactive, which means anything we do in our lab, we know the numbers got to get better when we actually send them to the Army lab for testings. So there is here, 50-minute half-life is fine, but there is good news and bad news. Good news, it works. Bad news, I think it's still too long. Then how can we deal with this problem? You could start thinking about the first material we dealt with is the benzene dicarboxylic acid. The porosity is in the low end. Second, either we are relying on the surface or defects, or if we are not, a carboxylate has to come off for the simulant or the agent to bind for the catalysis to take place. However, if we go to lower connectivities, then at that point, you have the active site already ready for the reaction to take place. And in the field nowadays, you could go make materials using linkers to dictate what node do you make. And you could go from 12 connected all the way to 4 connected. And I'm only putting the even numbers because that's the ones you could actually predict. Nowadays, you could make a 7, a 9, 11. I don't know how you could actually predict those. We got the 7 recently, and it's serendipitous. We couldn't have design rules for making, at least in my group, I don't know how, for a 7 connected. We made it by accident. But that's now, at least in the even numbers, you could actually now precisely predict these materials. And we were able to make the 12, the 8, and the 6. You have the UIO 66, NU 1000, and MOF 828. Now, if we look at the catalysis, that's what you see. It's quite incredible. You go from a 50-minute half-life to a 15-minute half-life, and with this 6, this is not kinetic traces. You can't do kinetic traces this way. But I wanted to show you which points going with which materials. But you could take 828, and your first point, at least with the NMR, with the fastest you could do, is a minute later, and the reaction is complete. That's exactly where you want to be. Now we have a reaction and a catalyst that is really, really doing quite well. But still, we are doing this in a beaker of water. That's not just in a dry, solid conditions. So how can we want to handle that? We want to do two things. The first thing, you have this ethylmorpholine there. That's not a good thing. That's a volatile base. We need the base, because if you have why do you need the base? So you have to think about when you hydrolyze toxic chemicals, what's the byproducts you are making? Because they could be just as toxic as the chemical you started with. If you hydrolyze a fluorine and F-, and if you have water molecules around, very quickly you could form HF. So HF, if you know anything about it, if you be inhaling HF, that will kill you just as fast. So you can't just say, I want to hydrolyze a toxic chemical. You want to also understand the reaction and what are you making and how can you capture that. And that's where the base is that will capture that actually very quickly, that the F- before it becomes an HF. And we need to replace that beaker of water. The first thing, let's talk about heterogenizing that base. And it was actually easier than I thought it would be. The first author on this paper, Chen, he was a grad student here at KAUST with Professor Adowdy, and he really did a spectacular job at taking these materials forward to where they are actually right now. The first thing, we heterogenized the base. So instead of ethylmorpholine, we used branched and linear polymers. And we got reactivity to work almost as good as the homogeneous ethylmorpholine. Okay, that's good. The second thing, how can we rely now on humidity? Let's go to the last MOF I talked about that has really the fast reactivity. If you go do a water isotherm of that particular MOF, you'll find out anywhere above 20% relative humidity, there is a huge uptake, that sharp uptake, and that's really just nothing but the filling of these cavities. That means anywhere above 20%, the cavities of this MOF is full. My gut feeling, this room is about 50–55% relative humidity. If I put that MOF right here, in no time it will suck up that water from the air, and the cavities will be full. If that's the case, that means I have a beaker of water inside the cavities. That means if my reaction is really taking place inside the cavities, I don't really need a beaker of water, at least on PowerPoint chemistry. That should work that way. But now let's implement that. But before we do that, we need to do two things. The question is, how can we put these materials in a mask? That's engineering 101. I don't need to waste your time on. If you make the right granulars, you could coat it there and you make a thin layer. Engineers know how to do that well. They don't care if it's a zeolite, activated carbon, or MOFs, they do that. This is a harder point, how you put MOFs on textile while you want to wash that textile, you want to be running in that textile, you want to be, as disgusting as it is, sweating in that textile, and it still needs to function, and it cannot just peel off. This is the suit that people use, and we cannot affect any of those layers except that black layer. That's the activated carbon, and we want to replace it with our material. So the first thing we need to do is put these MOFs on fibers, and we are not the first group to do that. But what we tackled in my group is we cannot use expensive equipment. We need a process that we could one day, if it works, we could process thousands of yards very quickly. That means it's not just a beaker chemistry, it has to be applicable. Even though I'm showing it in a beaker here, but I hired, before this project, I really did not know that there is people specialized in textile chemistry. So I hired a postdoc who did his PhD in textile surface science. And my God, that kind of science is complicated, but it's beautiful. He came and he actually did all this chemistry by being able to take our now polymer, the MOF, and we do this roll to roll. We adopted the same technology that our denim or jeans, how they coated and they put that color on the denim and the jeans. So that means, if you could do that with, I don't know how many millions of yards per year they produce, that means that technology exists. And we did that, and you could see before and after the coating. Okay, all this is nice, but does it work? And if you take now everything here, there is no beaker of water, there is no volatile base, and what we get a half time of 12 minutes, everything in dry. That's great. But one thing we've been told by the Army labs, for them to actually adopt and commercialize this material, it has to have a half-life with less than 60 seconds. It has to have a half-life less than a minute. Regardless how you do it, 12 minutes, there is no plus or minus 11 minutes. So that was about 11 minutes farther away. And because everything we do, we give it to them. This data from their labs, that's not my from lab. So they know exactly what data they are talking about. So one thing we decided to do, as I said, what I'm showing you today is very simple chemistry, because if you really one day you want to take something from the bench to the market, it has to be scalable, cheap, and you could actually do it. So every step you do, it has to be simple. What we decided to do is to take our branched polymer and actually link it or cross-link it with more hydrophilic groups. Why do we need more hydrophilic groups? We want to invite more water molecules to be in close proximity to the active site. More water molecules, that means your reactivity would be higher, because that's the reagent that you need. I could do this chemistry, which you could buy it from Aldrich, 4% cross-linking very quickly, and that's what you get. Once you cross-link it, you get that goo, hydrogel. When you mix it with the MOF, you could make that powder. But more importantly, you could maintain its porosity and its crystallinity. That means we're not blocking, because that's the main thing. When you coat and put a polymer or a hydrogel on a MOF, one of the things you are worried about is things are blocking the entrance to your materials, and then you lose the effectiveness of that material. And in this case, we don't. Kai, in this case, we told him to go and just test it before we go put it on fibers and textiles, because that takes time. Does this work? Your product is yellow, your starting material is clear, and what he showed, as soon as he dropped the starting material and it touches the solid, it turns yellow instantaneously. So we knew now we have really a composite that is very quick to do. It doesn't require a 12-minute half-life to do that. This is almost instantaneously. And at that point, we went and we put this on the textile. Once again, we maintain crystallinity and porosity. More importantly, when we do the catalytic reaction with the simulant, so you could imagine with the agent is even more faster than this. And now we are everything in the solid state, and we have a half-life less than a minute. So at this point, somebody, a professor at Northwestern University, you say, what else can I do? Because I very much got to the point I could get it to this point. And I will tell you later on where we took it from here. But there were a few questions that we really weren't happy with the first study. Not because it doesn't work. I am a professor. I need to understand certain basic science, because without understanding the basic science, it's very hard to bring something to market. Or how can I bring the second and the third generation to market? That requires both the basic science and the applied science, that they work simultaneously. The first enzyme required 2 zincs, and we said, okay, we have 2 zirconiums. Is that really a real requirement? Or nature was lazy for some reason, or it had a completely different reason why they needed 2 zincs. The second thing, is zinc the best material or the best metal for this reaction? Because zinc is plenty of it, and maybe that's why nature used it, but could be there is other metals that are better. The water molecules on that active site has to come off for the agent to bind. So what is the effect of that water exchange rate that on that active catalyst? And lastly, for the people who do enzymology and enzymatic reactions, product inhibition is something that a lot of times biologists have to deal with, because you make the product, nature knows how to get rid of it out of the active site, but here we don't know how to get rid of it very quickly, which means can the product kill the reactivity of our catalyst? And if that's the case, how can we fix that problem? And we need to do that with the same family of material, because if you change a lot of variables, then of course you're going to get different outcomes for not the right reasons. Here we wanted to stick with the same platform. The first thing, we found this enzyme called carbonic anhydrase. So if I zoom in into the active site, you'll see it's made out of one zinc. Okay, zinc comes back again. It's not a copper, it's not an iron, it's not a manganese, zinc. But it's only one zinc. And you could see it's linked to 3 histidines with an OH. One thing my team missed really badly. This paper was published in 2010. If you look at the reaction on the left side, that's exactly the hydrolysis reaction we want. But in this paper, they concentrated more on the, the structure of the enzyme, and they threw that reaction in the SI that we missed. So hopefully in the future, maybe, I don't know, AI or something can help us find things even in the SI, not just, I am guilty of it as well. I didn't read the SI, so we missed it. But that's also told us one thing. Maybe 2 zincs is not a requirement. 1 zinc does the job. Now let's go to the lab and make a material that resembles this MOF, and this is the MOF we decided to go after, MFU4L. Now if you put those 2 active sites next to each other, you could see there is a lot of similarities. But one thing we decided we could do that maybe nature wanted to do and couldn't, or nature did not care about doing, is, okay, can we replace that zinc with other metals? And we decided, okay, for now we could use zinc, copper, nickel, plus 2 transition metals. Very simple. The X in the active site of the enzyme is an OH. But why can't we put chlorides or nitrates or other formates, other anions? What is the effect of different metals? And what is the effect of other anions, coordinated anions, non-coordinating anions on the reaction? So we made this material. You make it with a chloride to start with. You wash it with a base. You make the OH, and you could see the morphology, the porosity, everything looks good. For the next slides, before I didn't put error bars on my catalysis plots. Now I'm going to start putting error bars because I want to make a point. So first of all, this is the blank. If you have no catalyst, there is some background from the reaction. You get maybe 1 turnover number after 60 minutes and stop. So it's good to know what is the background even if you have no catalyst. Now you do the zinc, and that's what you get. It's actually a good catalyst. Except if you pay attention to that plot, even though it's way high up there, you will see once it reaches around 92% conversion, slows down drastically, even get to a point that plateaus. To get to a full conversion, it might require another 6-7 hours. So it goes up very fast, then plateaus. And we wanted to understand, why is that? But at that point, we had a really hard time understanding by using just the zinc alone. So what we did, we decided to go and make now the copper, nickel, and the cobalt of these materials. And now you could see the porosity and the crystallinity, everything looks good. And now let's go to the catalysis. I showed you the background, I showed you the zinc. Now you could see the cobalt. The cobalt is an awful catalyst. You get couple turnovers and it dies. And it's not because it's degraded, because we could take the material, crystallinity is there is no metal leaching, there is no ligand leaching, the porosity is great, it's just an awful catalyst. Okay, you say, okay, let's go to the next one. You go to the next one, the nickel, so the cobalt was awful, the nickel is a tiny bit better, but not much better. So you might start thinking, wow, nature got it right. Zinc is the optimal metal until you do copper. You could see copper is really a fast catalyst, but it doesn't plateau at 90%. It reaches full conversion as fast as well. That means the zinc plateauing at 90% is not an experimental error. There is something there that it wants to teach us. If we don't do this fundamental study, we would have agreed, okay, zinc is great, let's make our 2nd generation from zinc and move on. But we wanted to understand why these 2 are bad catalysts, Why the zinc is very good and the copper is excellent. Okay, before I tell you that, let's take the copper, which is the best catalyst, and now make it with different anions to see does it make any difference. You do that and you do the catalysis. I always tell my students, you know, when you see a plot like this, that's more similar than different. So they don't make a lot of conclusion out of that plot. So the OH or the chloride, they're all good. Take those. Now let's go back to our different metals. You know, every once in a while, I don't like it, but I teach Gen Chem. And when we start thinking about this project, I'm glad I taught Gen Chem. Because the first thing I wanted to do is how can we analyze what's going on? Then everything is happening there is the water molecule coming off, the substrate is binding, the product is leaving or it stays binding. We wanted to understand all this, the water exchange rate. We wanted to understand what is the binding energy of the substrate to the active site. What is the binding energy of the product to the active site? Is it better or worse than the substrate? Is there a competition? Okay, how do you do that? Let's do titration. Simple chemistry. We did, and we have like an automated titrator. And once you titrate, what do you see? Our zinc MOF is very much similar. So like the first one is 7.5. The pKa of the water molecule sitting on the active site is very close to actually the zinc sitting inside that active site. That's already told us that we actually did resemble what's inside that active site. But if you look now at the copper, it's much lower. If you look at the nickel and the cobalt, it's much higher. Okay, that tells us why that order is appropriate. That means the copper is a better acid. It works better. It makes sense. But it didn't tell us anything why the zinc plateaus at 90% and why the copper and the cobalt are off for catalyst. Now let's do a different titration. Now let's do isothermal calorimetry. So how are we going to do it? We want to try to understand the binding energy between the substrate and the catalyst and the product and the catalyst. That means we make the product intentionally and we actually titrate it. And using this technique, we could get delta H, delta S without any assumption. Experimentally, we could extract it. These techniques used by biologists all the time in order to actually understand the active site and the binding energy of substrate to the active site of the enzyme and the protein, especially in the pharma industry, to see if there is a drug binds to the active site or not. But also, you want to, when you do this kind of titration, you want to make sure the background is not getting involved, and that's what you see. We want to make sure our buffer, our water, everything is not interfering. There is nothing there. And now when we titrate our product to the material, you could see these beautiful curves, and we could extract heats really easily out of these materials. To cut to the chase, that's what you get. You get, if you do a water exchange rate versus delta H, or if you do a pKa versus delta H, you see different plots, but in different directions. But if we take this plot here, this is telling us that the cobalt and the copper likes to bind water really strongly, but even when the substrate competes for that active site, they like to bind the product very strongly. That means the substrate will have a hard time binding. The zinc, it requires, it's somewhere in between, which means once you reach a certain concentration of the product, now the substrate will have a hard time binding, and maybe that's why it plateaus around 90%. The copper, it does not care about the water, and it does not care about the product either. And that's where Young-Teller distortion, if we learn it in inorganic chemistry, comes to play in our favor, because that twisted coordination is actually helping us not to allow that coordination to take place. So I hope I showed you so far that the first part of the talk, I kept the same metal. I changed the number of coordination around that metal. And in the second part, we kept the same ligand, but we changed the metal. And now my team really trying to cross between both techniques in order to make now Gen 3 even better than Gen 1 and Gen 2. But once again, without doing this basic science, we would have a hard time doing that. Just to show you a little bit about a collaboration between, myself, UT Dallas with Jeremiah and Harvey from the Army labs. So they took the enzyme encapsulated in our MOFs, and they put it under, I'm not a biologist, so I don't know what they do. They put it under by the fat layer. It's not injected, but they drop it by the fat layer, and slowly it diffuses into the blood. And here they are doing two things. If the enzyme is encapsulated or not encapsulated. And those enzymes are actually been discovered by the Army lab and the Weizmann Institute to be actually a good antidotes for nerve agents. And we see, yeah, they could give you protection for a few hours, but is not enough to last for a long time. And we showed in our materials, you'll see in a second some plots, that we have actually protection for almost 82 days. And the second thing is you want to know, I've been taught by my biologist friends that you want to see that your body is not reacting against what you are injecting. And what you see here, actually with our material, which is, you could go to almost 60 days and you see multiple injections and you see that really your body is behaving the way it's supposed to behave. Versus the ZIF-8, it has after a few days, it starts to actually behave a little weirdly. And when they do this now with mice, we could go up to about, as I said, 82 days with full protection. Once again, this is animal studies, doesn't mean anything. That's what I learned from biologists. That means it requires many more years of actual testings before you actually make other conclusions, just to say that these are very promising results. All the technology we learned, how to do it in a mask and a textile, now you could make it into the colloidal, not injectables, that things that you could drop in a certain layer in the body, and they do some well. So these pictures, sometimes they make me dizzy. But you could see. So we started with talking about a beaker, and now we talked about biology. But the first project I told you, now we're making textiles to a certain point, and now somebody has to take it to the next step. And one of the companies, I was the co-founder of, and still the chief scientific officer, is called Nomad Technologies. We started it with my colleague, Ben Hernandez, with the idea to actually answer 3 questions. We did not know where we will be 10 years later, and I'll tell you where we are 10 years later. I am sure a lot of people in the audience, including Muhammad, will understand this when we were giving talks a few years ago. We'll always get the first 3 questions. Let me tell you what the first 3 questions. MOFs are not stable. MOFs are not scalable, and they are damn expensive. You get tired of having to answer those questions over and over again, but they might be right. And the only way to test it is to really do that exercise, because buying things from Aldridge, obviously everything is expensive, but you needed to do it at the right scale. So we started this company, and we wanted to have enough people in that company that they only concentrate on one material, and it's MOFs. They are not doing MOFs, zeolites, activated carbon. No, right now we have about 80 to 90 people. That's all they do is MOFs. And the second thing, if you really scale it up to a certain point, how can you also reduce the impact of that scale? And right now, the scale, we could run reaction about 3 metric tons. So that's 3,000 kilos per reaction. And I will show you some of those pictures. But these are some of the materials was produced. I always say making now a 1 metric ton of a MOF is not a challenge. What's the challenge is, what do you do after that? Being able to filter a 3 metric ton MOFs, being able to remove the guest solvent from the cavities of 3 metric tons while maintaining the quality control. You want your 3 metric tons and your 1 gram to have the same thing. That requires the right people to do this, and I will show you who are these right people. And then you got to start working with customers. And we do work with customers all the way from design to testing to actually partnering with our partners to put pilot scale plants in their facilities, and I will show you some of that. I'm going to talk about 2 product lines. There is a third one. We'll leave it alone for today. But the first, you have INX, which is for the semiconductor industry, and the Sentinel is for clean air or, let's say, toxic chemicals protection. The first thing is INX. We all have our iPhones and iPads. These came to be working really well because the Intels of the world, the Samsung of the world, they have to take that wafer and dope it with a boron or a phosphorus, and they use BF3, arsine, phosphine. Those are really toxic chemicals. And they typically use gas cylinders up to 20 bar, and anytime there is a leak, that actually they have to evacuate the whole fab, and evacuating the whole fab, assuming nobody got injured, because these are incredibly super toxic gases, that costs millions of dollars just to evacuate the fab. So what we decided to do, we decided to come up with the idea, can you make MOFs that can store as much below subatmospherically, as much as the empty cylinder at 20 bar, but now because they are subatmospherically, if there is a leak, the air will come in and the toxic chemicals would not go out. So you have same capacity, but you gain the safety, and then you have to reduce the number of shutting down your facility. So we have now these 4 products, and this product called INX, one for BF3, arsine, phosphine. And this actually right now is distributed by Merck Germany since 2017, 2018. They are now being used by 42 fabs around the world. They are used in Japan, in South Korea, in Taiwan, in Singapore, in Europe, multiple places in the US. In 2019, we opened our first facility, which is in South Korea. So we actually make those cylinders in the US. We ship them to South Korea. They go, they get filled by those. That's where Merck Germany is. They fill them with the gases, and they distribute them to Japan, South Korea, Taiwan, Singapore. So that's the first product. The second one is the Sentinel, which is related to what I talked about in the first part. That's where NUMAT and the Department of Defense collaborated, and they scaled this material. They actually integrated. You could see you don't see that, but trust me, all these suits, it's enabled by a material that has that. I want to show a video. I think I still have some time. Show a video, if it's, quickly. It's been bought by the Army, so what I'm showing here is out there on YouTube. I'm not showing you anything I'm not supposed to. But everybody on these videos are actually wearing suits enabled by a MOF. That's amazing, at least for me. So you get to the point. Basically, they wear them, they put them to use, and then they test them to make sure actually everything is working, and everything was working just the way it's supposed to. And they put an article, everybody can go and find it and read it, about this technology, showing that these suits are light, they are breathable, you could run with them, which the old suits you can't, and that's why soldiers did not want to wear the activated carbon-based MOFs, because the layer of activated carbon was so thick that they are so stiff, and in no time you start sweating, and the soldiers will almost faint, that they did not want to wear them, versus here, everything was supposed to be. And now, we've taken them all the way, as I said, beyond defense into other areas. For the other 2 facilities of NUMAT, we have a facility in Wisconsin by Green Bay, and that's where we actually manufacture these MOFs. Then we truck these materials into Humboldt Park, Illinois, and you could see that whole entire campus is a newly, maybe 2-year-old renovated campus by NewMat. Let me tell you just one slide about this campus because I was excited about when I learned that myself. This campus was built in 1906. In 1920s, 25s, and 30s, Nikola Tesla worked there. And his 100th patent was discovered in that facility. And in the 70s, they shut down this facility in South Chicago, and it got like, basically was sitting there. It was nothing, disgusting. And when the agent took us there, the real estate agent, she thought we will never go for it. And when I learned about that story, we said, this is exactly where we need to do science. And we took this facility and we invested tens of millions of dollars to renovate this facility. You could see before and after the outside in this case. And more importantly, what you see inside, these are the pilot. This is a drying unit. That's how you dry a 3 metric ton materials. These are pilot plant reactors that they go all the way from 1 liter to 10,000 gallons. You couldn't do that by hiring a bunch of chemists like me. It requires more than that. It requires the chemist, the chemical engineers, the material scientist. Until 5 years ago, I actually did not know that there is control engineers. If anybody is a control engineer here, I apologize. But they are amazing how they integrate everything to work the way it's supposed to. And we hired people not just from academia. We got the people from Grace who've been formulating catalyst for pilot plants and from air products. Those people were running pilot plants that we brought them in to teach us how to do that. Last slide. I hope I don't offend anyone here, but I like to show this slide because if you go about a hype curve, every technology has the same hype curve, the same shape, except the time scale is different. I have to say, I'm so happy that this field just a couple of weeks ago won the Nobel, which is an amazing thing for all of us. But also there is one thing important. What happened right there for us to start going upwards? And I have to venture and say that this product had a lot to do with it. Because for a while, people start thinking, okay, a lot of people, MOFs been around for a while, which that's not true. MOFs are a young field, but people had no patience. People seemed like, at least government funding agencies, they thought, you guys been doing that for 15 years, and they don't understand. 15 years is actually nothing for a material discovery. But they changed their mind when NewMat actually was able to commercialize this all over the world. With that, I want to thank you and happy to take any questions.
Brilliant talk, brilliant talk. Very nice. Can we have questions? Right, start with.
Thank you, Professor, for the presentation. I just have one question. You said for hydrolysis to be activated, it should have 20% humidity. What if humidity is lower during fires?
Yeah, I technically never told you which MOF inside that suit. Correct? Okay, we had another program with the DARPA, which is for really harvesting water in a desert area for special forces. And we are not, in that program was my team, few other teams, Professor Yagi's team as well. And we actually found some MOFs we could harvest water brilliantly down to 5% relative humidity. So I showed the one that we could publish and talk about, but maybe not the one that is integrated in a suit. But you're absolutely right. It needs to work in a dry desert environment, or if you are in Alaska, it needs to work just as well. Yeah, thank you for your question.
All right. Yeah.
Thank you very much, Professor Omar, for the talk. It was amazing, but there's one of the slides you talked about how you prefer to use half-life over the turnover frequency. Can you please go over that again on why you preferred it?
Yeah, you know, turnover frequency, we could talk about it. That's easy. The turnover number is the issue. Turnover number, it depends on how long you run your reaction. You gotta assume everybody runs it the same way. Yeah. Versus a half-life, if I run my reaction for 2 hours and you run your reactions for 1 hour, the half-life will be the same. And the Department of Defense decided they want to use half-life this way regardless where we are in the world, and doesn't matter how long we run our reaction, as long as you have enough data points, you could extract the same number.
Do you feel like this could be a metric that should be used more often in chemistry? Or do you feel like it just worked for you?
You know, again, I think all metrics are important, turnover frequencies, turnover numbers, half-lives. It depends, as long as you standardize things, yeah. Because the main thing is to compare and contrast my results versus somebody else's results in a different lab, in a different continent. Yeah. As long as there is a way to standardize that, I don't think it matters which metric you use, and I don't have my favorite. Everybody has their favorites, but I don't.
One more question, I'm sorry. But in one of the slides, one of the byproducts were HF?
Correct.
Was that just like an in-lab test, or was that?
No. I mean, when we want to balance an equation, yeah. You will generate HF, and that's why we need the base to capture the HF. So your catalyst will hydrolyze the agent. Then you form an HF, very toxic, that you need to capture too. And the rest of the molecule, not so much. You don't care much about it. And that's why the base, that polymer that we use, so the polymer ends up doing two things. It's your binder to bind your your particles of a MOF to the fiber, and it's also your base to capture that HF from seeping further.
Thank you so much.
Of course.
All right, let's move on to another question. There, Vincent.
Thank you so much for the talk. I have one question about metrics again. So you were showing first a new 1501, which has tremendous porosity, and then I was going, and some of the materials you showed, like the LVT MOF with zirconium, I think it has like much more limited porosity.
Sure.
What is the actual porosity your materials need to address what you're trying to do, or do they even need to be porous, or like can surface just work?
Good question. Yes, they do need to be porous, because if you just rely on the surface, your active sites are limited. And that's why monolayers would not work here, because and we test them in the Army labs, what's a monolayer versus three-dimensionality. Porosity is important from a diffusivity point of view. The larger the apertures and the cages, the faster diffusivity. But you don't want it to be too fast of diffusivity. And why is that? You want the catalyst to keep up with your diffusivity, because if you're diffusing much faster than your catalyst can detoxify, then some of your molecules will reach to the other side as active agents. And that's why you have to balance what's your diffusivity rate versus your catalytic rate. All the numbers I showed you here, I didn't talk about that, but it's very crucial for you to make particles of different materials that they are in similar size. Because the particle size and diffusivity, they have, the smaller the particle size, the faster things can diffuse versus large crystals. So when we compare and contrast different materials, we try to make all these MOFs have similar particle size. Otherwise, your numbers will be all over the place.
Thank you.
Yeah.
All right, one or two more questions.
Thank you so much, inspiring talk. Just a quick question. You mentioned, you touched on AI. Where do you see like acceleration, or have you integrated this kind of tool already in the work?
Not in my lab at Northwestern, but yes, at NUMAT. And the reason for that, because in 2016, 2017, our software engineers decided to do something that I never understood why. They decided they want to connect every instrument that we use and be able to save every isotherms, every PXRD, every reaction we ran in a specific format. And I thought that just waste of money, waste of time, except right now we have large companies, I can't mention the name of those companies, are collaborating with NUMAT, not because they care about MOFs, not really. They care about the data we have to train their machines, because we have hundreds of thousands of data that we've been saving since 2017. And personally, I did not know what to do with. My group at Northwestern, we really don't do that. But as I said, the software engineers did it. At that point, I think they wanted for an easy search in the future, no more, no less. Now we find out there is actually more to it than that. So we get really, I would say, two large companies or entities that they've been collaborating with NUMAT for a while just to train their machines.
Okay, is there any last burning question? If not, okay, there's one here. Yeah, I can just hold it here.
Thank you so much for your talk. So I have a question about the zinc and the copper. So since the different percentage, is it better to use, like for the longer period of time, to use zinc or copper? So is this small different percentage affect the usage of metal over time?
Maybe not, but when you see something that doesn't make sense, you want to understand why. And that's why still doing fundamental science is crucial. And right now, if I want to say what's the next generation's material should be, I would say copper, because I am guaranteed that in 10 minutes, everything will be detoxified. Versus the zinc, it might take hours before that reaches that. That might be okay for certain applications, but at least you have the optionality to choose a zinc or a copper based on performance and cost and everything else.
All right, I'm going to ask the last question. There is Sultan again.
Uh-oh. Yes, sir. I don't see why not. But are you, at that point, you just releasing oxygen only?
No. So I'm a technical diver, so I go very deep. And
okay.
Sometimes there are a lot
so I am not a diver. I can't even swim. So you have to educate me about what a diver breathes.
So besides commercial diving, technical diving, you just decompress for a while. But besides that, you wear a dry suit. So it protects you from a lot of the biological stuff inside. But I don't know about the gloves and other things. Also, breathing underwater, that could be an application. I don't know, but I saw the fabric.
Honestly, I don't know if that would work underwater or not. One thing we found out that the suits actually work better when somebody's sweating, because actually the Army was worried about, because once you put the suit on, that means there is an alarm. When you get an alarm, it doesn't matter who you are, you'll be sweating. And we found that our reactions are actually working better, because sweat comes with moisture. The more moisture you have, the better your reaction. So underwater, I don't know. Maybe that's our beaker.
All right. Thank you so much, Professor.
All right. So I'm going to.
You had a question.
I had the last question.
Yeah, you had a question.
Speaking of science here, what made you jump from boron clusters, your PhD at UCLA, to MOFs? How do you? What's the advice here to the young people, the students?
So okay, he's asking me. Okay, my background for my PhD, I am an organometallic boron chemist. And I made boron clusters, which you could think about them as three-dimensional aromatic benzene rings. Okay, three-dimensional.
All right.
So I went from small molecule three-dimensional materials into materials three-dimensional materials, which now we are incorporating my three-dimensional ligands into the three-dimensional MOFs.
You got fascinated by the
Exactly.
3D.
I like 3D.
So, okay, so how does the young people take it? Is it like they're working on something they really like, and then they just look ahead. And
exactly. I mean, you have to do what you enjoy. I mean, I love the MOF world because it makes sense to me. It makes sense to me mathematically. It makes sense to me from coordination chemistry. The sky is the limit in a way, and there is a lot of open space for a lot of people to play in that playground.
All right, with that wonderful note, let's thank Professor Farha again.